Hi, everyone, and welcome back. This is our fifth and final lecture in the special series on the biomolecules of the SARS coronavirus 2 virus that's behind COVID-19 and spoiling all of our plans for the summer. So the last biomolecule that we're going to talk about is it's literally at the heart of the virus and at the center of a lot of the biology that if you go on to study biology, you'll be interested in. And these are nucleic acids. So even though they account for only maybe 1% of the total mass of organisms, the information that they encode is responsible for all of the structure and the function and the look and the feel of the organism that builds out of it. So when we talk about nucleic acids, we're looking at two major molecules here in terms of macromolecules, polymerized nucleic acids. So this would be ribonucleic acid, so RNA, and deoxyribonucleic acid, DNA, which are polymers of nucleic acids. So what does that look like? So here we have the 5 nucleic acids that are present in humans and in most life on Earth. So adenine, guanine, cytosine, and thymine are the nucleobases, the 4 molecules that make up the bases that are the monomers of DNA. And for RNA, we replace a thymine with uracil. And those bond together as shown in this other picture here, where this is an RNA because it's using the uracil. So each of these nucleobases, these nucleic acids, consist of a sugar backbone. So this is a ribose, as in ribonucleic, connected through a phosphate to the next ribose. So this is the backbone of the RNA. And each of these side groups that carries one of these nucleic acid monomers bonds with another one either through 2 hydrogen bonds or 3 hydrogen bonds. And this is why we always hear about these things pairing up. And it's because either it will be able to bond with the 2 hydrogen bonds or the 3 hydrogen bonds. So let's have a look at what that looks like in practice, where we've got a double-stranded DNA and here a single-stranded RNA. Again, these nucleobases are going to be colored in accordance with what's shown on the side here. And the backbone is the ribose with the phosphate connecting them. So in all cases, you'll see the red is joined to a yellow, so this is the thymine and adenine, or a green with a blue, so cytosine and guanine. And that has to be consistent again because it's going to either bond with 2 or 3 hydrogen bonds. And the order in which these CGAT letters form the long chain of the DNA strand or the RNA strand, that provides the information that encodes how to produce proteins, which are really making up everything about us, as we've seen in a past video. So all of the structural proteins, all of the functional globular proteins, as well as all of the membrane proteins, are built up in accordance with the information that's stored in the DNA and usually transferred through the RNA. So how does this work in practice? So when we talk about DNA, we also talk about genes. Genes aren't really separate molecules by themselves. Think of them more as regions on a DNA chain that are encoding for specific proteins. So where if there's a call for a new protein to be made, that section of the DNA strand, that gene, is going to be, let's have a look here. So it's going to be opened up by polymerase that will decouple those 2 strands of the DNA and read off of one of the strands to produce an RNA that can be transferred away from the DNA, so out of the nucleus where we store the DNA, and into the rest of the cell where that RNA will be read in order to make the protein. This is exactly like if you had some master plans for some widget at a factory. You wouldn't necessarily want to go back to the master plans, bring it out to the shop floor in order to plan out how you are going to produce that part. You might go to the master plans, make a photocopy, and then read the photocopy out on the shop floor. Well, that's exactly the same thing. That's what's happening with this RNA polymerase. Opening up the DNA, reading one of the strands, and using that to produce an RNA copy of that strand that can then be sent out to produce the protein. Okay, so what happens to our RNA strand, so this messenger RNA, as it leaves the nucleus, or after it leaves the nucleus, is going to be read by a ribosome that is going to work with transfer RNA in order to build up the proteins. So every three nucleobases will encode for one amino acid. And as it's reading through these set of 3 nucleobases, the ribosome is collecting together the correct amino acids in the correct order to produce the primary structure of the protein, which will then be folded into the secondary and tertiary and quaternary structure of the protein in order to produce whatever function that protein is supposed to do. Afterwards, the messenger RNA can be broken up and reused. This is exactly where the coronavirus is going to take over the rest of our system. So the SARS virus, the coronavirus, is a RNA virus. So that means that inside of its envelope, it contains an RNA strand that when it bonds to our cellular membrane and its surface proteins open up that receptor in order to allow its RNA strand to enter our cell, the ribosomes don't really care the source of the RNA. So as long as it's getting an RNA that it can read, it's going to start reading it through and building up the amino acid chains as instructed. So as soon as that viral RNA is introduced into our cells, it can start working on the translation of its RNA code into an amino acid chain, which will then be packaged through the rough endoplasmic reticulum, and then the RNA will be replicated, packaged up inside this group of our lipid membrane that's now been decorated with all of the proteins that its viral RNA has encoded for, packaged by the Golgi apparatus into a vesicle and ready for exocytosis, or kicking it out of the cell. So this is the viral release that then goes on to infect other cells. And this is happening thousands upon thousands times per cell. And that is how we get the growth of these virus inside our bodies. So that's the general process. But let's have a look at what this viral RNA sequence looks like. So one of the things that we know how to do actually pretty readily right now is to read exactly what order of these nucleobases forms the RNA. So when we take in a new virus or a new organism at all, we can have a look and see what order all of those nucleobases fall into. And this gives us the viral genome. So this organized set of letters, so A, T, T, A, A, G, G, T, T, is the strand. So this is what what those ribosomes are reading off to produce virus. And you can look this up on the National Institutes of Health has quite a good database of the viral genomes if you're interested. But this is one of the original ones that was submitted in December 2019 from China that's going to help the world get a handle on exactly what proteins are being made, looking for any weaknesses in this genome that we might be able to attack to either cut up the RNA strand and interrupt its self-replication, or give us hints about what we can look for inside the proteins as a target for either an antiviral drug or ways of replicating it that will allow us to build a vaccine in good time that's going to be effective against the virus and against nothing else. So here we go, still looking through, and this is a short, small genome. And we have all the way down here at the bottom, almost 30,000 nucleobases in this, what is a relatively short and simple RNA strand. So at 30,000 bases, that means we're building about 10,000 amino acids into about 10 different structures. So some of them are going to take more of that than others, but this is what we're encoding for. The other thing you might have heard about or been wondering about when it comes to the genetic sequence of the DNA is mutation. So as an RNA virus, it doesn't have that second strand that a DNA has in order to be able to compare copies. The one that's being copied and like a verification strand, because they have to pair in certain ways, you can always check your copy against the original when you have a DNA to prevent any accidental replacement of nucleobases. Because when we talk about mutations, that's really what we're looking at, is just the random accidental replacement of what should be, say, an adenine with a thymine, or et cetera, et cetera. RNA doesn't have that extra layer of protection, so it does mutate a little bit more easily. Does that mean it's going to get worse, better, or indifferent? Well, most of these mutations are going to be completely neutral. They're just not going to matter. Or they're going to be harmful for the virus. If this is such a finely tuned virus that it's as infective as it is and as deadly as it is for people, any change you make to that, if you just make a random change, is likely to be worse as a virus than it is right now. Especially if there's no evolutionary pressures to make it better. So something that might be more transmissible is a mutation that it's more likely to keep. Something that would make it much more deadly, where the host that has that mutated virus is just more likely to die before it can spread that mutation to a different host, is not as likely to survive. However, this virus has mutated many times, even in the few months that it's been circulating. There's a great resource for that that exists, where we're looking at the genomic epidemiology of the coronavirus. And we can actually watch, let's see if we can. Yeah, we can. We can watch this virus as it leaves China and starts to mutate into Europe and around the world. So let's see if I can move my face. I can. Okay. So on the map, you should be able to start seeing it grow in Asia. And as the different strains of the virus appear in different countries, it's going to have small mutations. So here, just encoded, we have the green ones are going to start showing up in the United States. And pretty soon we're going to have orange ones, showing up in Canada as well. So here's a few Canadian mutations. So there's, it doesn't say what province it's from. This one's from BC. And we're likely to get some Ontario. Okay, so here's a genome that was submitted from Ontario. Again, with small mutations as it goes forward. So there's one that led from it from New York. Here's some more Ontario strains. And we're going to be able to track those just because of how many mutations, so how different that is from the original strain. And this is going to keep keep changing and keep going over time. So this is a dependence on not just which biology is more successful, but what areas are going to be, what area is it going to spread more? Because with more hosts and more replicated copies has more opportunity for mutation. So what we're seeing here isn't just a biology chart, it's also a political chart to say which areas are going to have more spread. You're just more likely at this point, speaking in April of 2020, you're likely to see a lot more mutations come out of the United States and the European ones come out of Italy, just because that's where more of the cases were. Okay, so that's going to sum it up for this short lecture series on the biomolecules, and especially as they relate to the coronavirus. So I hope you found some of that interesting. If you want to talk about this or any other subject, you can always look me up, and I'm happy to talk about that. Remember, none of what we talked about over this whole series is going to show up on the exam, so you don't really have to worry about that. It's really just more for your interest. If you are interested in any of this, RMC offers biology courses through the chemistry department, and it's available as a life science minor. So if you're taking one of the other sciences, you still can learn more about the biology, both in lecture classes, and we also offer labs where we do microbiology all the way up through ecology and environmental sciences. So I hope you got something out of that, and I guess we'll see you next time.